হ্যালো
স্যার।
স্যার, ম্যাডাম আপনি আমাদের বাইরের যারা ডিস্টিংগুইশ স্পিকার ছিলেন বা এক্সপার্ট যারা ছিলেন তারা জয়েন করেছেন?
তারা দুজনেই জয়েন করে গেছেন।
বাস, ওকে, আপনি শুরু করুন। আপনি শুরু করুন।
স্যার, আপনি অডিবল এন্ড ভিজিবল বোথ?
এভরিথিং ইজ ওকে, ভেরি গুড। আপনি শুরু করুন, অডিবলও এবং ভিজিবলও।
A very good evening to everyone. On behalf of Zoology Department, Shabong Sajanikanto Mahavidyalaya, I welcome you all to this international webinar entitled Pharmacotherapeutics of Diabetes and Epigenetics of Hypertensive Nephropathy, organized by our department in association with Internal Quality Assurance Cell. I wholeheartedly thank all the participants for being a part of this webinar. Hypertension and diabetes are two chronic diseases believed to be responsible for highest morbidity and mortality worldwide. In this asphyxiation of present COVID-19 pandemic, all activities of human civilization seem coming to a halt. Even in the present scenario, the issues of chronic diseases like diabetes and hypertension are very much relevant because these comorbidities contribute significantly to the death and poor prognosis of infected population. So, the topic of today's discussion will definitely throw some light to the gray zone of these two chronic diseases.
Madam, something is wrong. Something is wrong, madam. There, the sound is not coming from you.
No, sir. No problem, sir.
Sound is not coming, sir.
No, sir. Actually, sir is having some. Maybe.
ঠিক আছে, সব ঠিক আছে?
I'm audible now?
হ্যাঁ স্যার, ঠিক আছে স্যার।
ওকে ওকে।
I, Dr. Shongita Shamadar, Organizing Secretary of this webinar, extend my sincere gratitude to the President of our governing body as well as Honorable MP of Rajya Sabha, Dr. Manush Ranjan Bhuiya, who is also the Chief Patron of this webinar. I also express my sincere thanks to our Honorable Principal Sir, Professor Dr. Tapun Kumar Datto, who always extends his encouragement and support towards these types of academic activities. We are proud to announce the presence of two eminent scientists, Dr. Partho Roy, Professor, Department of Biotechnology, IIT Roorkee, India, and Dr. Utpal Sen, Associate Professor, Department of Physiology, University of Louisville, USA, as distinguished speakers among us. I am specially thankful to them. I express my thanks to Dr. Selim Chisti, Convener IQAC Shabong Sajanikanto Mahavidyalaya. I am very much thankful to the technical support team. I convey my thanks to the advisory committee and also the faculty members of our department. I am pleased to inform you that today in this digital platform, we have a good number of participants, not only from West Bengal, but also from different states of India and abroad. Now, I would request our principal, sir, to inaugurate the session. Over to you, sir.
Thank you, madam. Thank you, everybody, and welcome everybody in this webinar. Pharmacotherapeutics of Diabetes and Epigenetics of Hypertensive Neuropathy, which is held today in our digital platforms set up by our Shabong Sajanikanto Mahavidyalaya technical team. They are arranged such a manner. This webinar is aimed to confer about most common silent but deadly disease, diabetes. I think it is based on the diabetic theme. Interesting thing is that the carbohydrate is our body is essential because it is transmitted into the cell from blood, converted into a simpler form, and takes part in the productions of energy through metabolic pathway. It is going on in our body in every sophisticated and also sensitive biochemical pathway in controlled manner. But if any uncontrolled or disorder in regulatory mechanism or regulatory pathway occur, then several problems, biochemicals, consequences resulted, and we feel several problems and various symptoms found. We actually everybody know that we eat mostly carbohydrate. Our body turns into sugar, transforms it into a sugar called we know it is glucose, and send it into our bloodstream. Our pancreas releases insulin, a hormone that helps moves glucose from our body into our cells, which use it for the productions of energy or produces energy in various biochemical pathways. Diabetes, when the metabolisms of glucose is we talking about, then two main things we imagine that in our blood a specific amount of glucose concentration found in all the times and maintaining all the times. And if by somehow its disbalance occur, then various consequences found, and commonly the diabetic symptoms that is generally occur. The reason is different. Our expert will discuss about its various consequences, various results. It is silent and deadly killer of the body. However, I am thankful to our organizing secretary to set up such a nice and very common themes for the discussions, and I am also thankful to our two distinguished researchers in this specific field, and I am also proud to announce this international webinar in these platforms, and I feel proud to honor to warm welcome our honorable distinguished speaker and resource person Dr. Partha Roy, Professor of Biotechnology, Indian Institute of Technology, Roorkee, Uttarakhand. He will talk about some chemiferol, a flavonoids that plays a critical role in protecting pancreatic beta cells by autophagy in a lipotoxic environment. And our another distinguished speaker, Dr. Utpal Sen, Associate Professor of Physiology, University of Louis Valley, School of Medicines. Inert, he will talk about the inert immunity, a crossroad of hypertensive neuropathy. Sir, I am also expressing my warmth and deepest gratitude to all in these occasions and waiting for your realizations over the issues, your research findings, and your contributions in this huge knowledge world. I must congratulate this to the organizers to select such a fantastic topics. I am also I am also express my thanks to all of the technical teams of this webinar. I would like to welcome our three advisory members of the advisory committee. One of them, Selim Khisi, Avik Mukhopadhyay, and Sunita Mitra. They are useful advice for the webinars. In this occasion, I am requesting to the organizing secretary to request our eminent resource persons. Sir, we are excitingly waiting for your treasurer's speech. Please welcome Dr. Partho Roy and Dr. Utpal Sens for the sessions. And as usual, before leaving the introductory or welcome speech, I must request all of our participants, those are present here in online, please mute all of your microphones and please don't present your screen because it will create huge disturbances to our distinguished speakers. Sir, welcome. Thank you all.
Thank you, sir.
Welcome. Okay.
Thank you, sir, for your warm welcome speech. Now I would like to welcome Dr. Partho Roy to deliver his talk. His topic of discussion is camphorol, a flavonoid that plays critical role in protecting pancreatic beta cells by autophagy in a lipotoxic environment. Dr. Partho Roy completed his bachelor's and master's in zoology from the University of Kollani, West Bengal, securing first-class first position both in UG and PG. He obtained his PhD from Vishwabharati University and then he moved for postdoctoral studies at Institute of Reproductive and Developmental Biology, Imperial College London, UK. Currently, he is a professor in the Department of Biotechnology, Indian Institute of Technology, Roorkee. Prior to joining at IIT Roorkee, he served as research scientist at Glenmark Pharmaceuticals Limited, Mumbai. His current research interest is development and validation of nutraceuticals for the cure and management of various diseases with special emphasis on diseases like cancer and diabetes. He has published more than 120 research papers in peer-reviewed journals and with high number of citations and authored several book chapters. He is the recipient of Outstanding Teacher Award at IIT Roorkee. Professionally, he is serving various scientific and academic bodies in India as panel members. He has visited various universities, institutes across the world as visiting faculty. Now, I hand over the microphone to Dr. Partho Roy for his valuable talk. Dr. Partho Roy, sir.
Okay.
Over to you, sir.
Okay. Thank you. Can you all hear me? I am audible?
Yes, you are audible and visible. Yes, sir.
Okay. So thank you very much, Sangeeta, for such a nice introduction about me, although I don't know if I deserve them all. Before I start my talk, I would like to convey my sincere thanks to Sangeeta, who introduced me here, and Professor Datta, Principal, sir. I am really honored to have this invitation from you all. Distinguished faculties of this college, as well as my dear students all over Bengal and outside Bengal. So it is my honor and pleasure to be here to talk to you about some of our work that we are doing on in our lab. So I will start sharing my screen. Okay, so today's talk is assessment of the role of camphorol in protecting pancreatic beta cells by autophagy in a lipotoxic environment. So if you see the topic, it involves a couple of things here. One, I am talking about camphorol. Camphorol is basically a flavonoid present in the plants, and protecting the pancreatic beta cells. As you all know, pancreatic beta cells are extremely important for maintaining and regulating the diabetic conditions. I am talking about one issue here is autophagy. Autophagy is a process of cellular organelles destructions. It has both positive as well as negative effects. So I am talking today about its positive effect on the cells. And here I have mentioned one term called lipotoxic, means here I am talking about what happens to a pancreatic beta cells in a lipid overdosed condition. What happens if the beta cells comes across a high amount of lipids in a cellular condition? So that is the whole topic of discussions will be, and I will show you some of our recent data that we have come out for this. So it will involve some research aspects and some of the fundamental aspects. So students, please bear with me, and after the talk you can interact with me wherever you find it a little bit complicated for you. So if I start with this, so what happens, this is beta cells, lipotoxicity, what happens there? So if you see, these are the fat cells in our body. We have large number of fat cells in our body. So when the obesity occurs, the fat cells swells up. So there's they enlarge in size, accumulate large amount of fats, and then because of the presence of macrophages, they tends to become bigger, and then they leads to several types of effects like oxidative stress, DNA damage, telomere dysfunctions, which ultimately leads to the activation of p53. And as you all know, this is basically a tumor suppressor protein, which actually is responsible for removing or killing the cells. Now, in the diabetic condition, what happens when this situation occurs in the pancreatic beta cells? It is not good. This is good for tumor or cancerous conditions, but other conditions, high regulation of p53 is not good because it actually starts to kill the pancreatic beta cells. Also, it pushes the cells towards insulin resistance and diabetic conditions. So that means high fat is directly linked to the diabetic condition. That's why we always say obesity-linked diabetes, which is a very serious problem, and it's quite difficult to cure. And the medicines that we have there, they provide some temporary reliefs, but they are not the exact cures all the time. So with this in mind, we started working on this. And so it's a major cause of obesity-linked type diabetes is because of lipotoxicity, because if the lipid amount in the body is very high, it leads to apoptosis and kills the pancreatic beta cells. So if we are able to prevent the apoptosis of these pancreatic beta cells, we can, to some extent, will be able to control the diabetes in the patients or control the obesity-linked diabetes. So this is basically, I am showing here some of the vegetables, fruits, etc., which are rich source of flavonoids. As you all know, flavonoids is a very important phytochemical constituent present in these fruits, vegetables, and so I am mainly talking about the nutraceutical aspects of it. So that's what our basic research is, that we try to look this food as a drug, not the drug as the food. So we the reverse. So we want that if you eat good food, you will be healthy and you can prevent many of the disease conditions. So what is these flavonoids are? So basically they are phytochemicals, and they have 4 categories of them: flavonols, flavones, and isoflavones. And camphorol, what I am talking about today, is one of the members of flavones. So this is one of the members of this family, and very important member. And this includes quercetin, myostatin as well. But camphorol is one of the critical one, which has got tremendous role in various health beneficial impacts. And it has some of these present in many of the food sources like onions, fruits, broccoli, apples, all sorts of fruits. They will have these. So it's a very common phytochemical and have lots of health beneficial effects. So this is basically natural flavonol, and it's basically comes under the family of flavonoids. It has got antioxidative, anti-inflammatory, anti-cancer activities. For about anti-diabetic and anti-obesity activities, not much information are available. So one of my students, Ritu, joined my lab, and she wanted to work on these aspects of diabetes, and she said that I want to work on camphorol. So she took up this camphorol, and as you can see, this is the basic structure of flavonoids, and this is the camphorol. Large number of hydroxyl residues are there, and this is the chemical structure of camphorol. And you can see different companies sells camphorol as in tablets because of its nutraceutical potential. It's very important health beneficial impact. So sometimes it's nutraceuticals or functional foods it can be used. So these are commercially available. So So we want to explore the mechanism of these particular phytochemical for the cure and management of diabetes. And then we wanted to link one more important phenomenon, what we call as autophagy. So autophagy, as you all know, is a process of self-eating. Self-eating means self actually destroys some of its damaged or destroyed or defective cellular organelles, macromolecules, etc. So this process involves the lysosomes and other cellular organelles. So lysosome is a key player in this process, which actually destroys the defective organ. So cell, if it is able to destroy its damaged or defective parts, so it will be easier for the cells to survive. So that is one of the process of cell survival process. So it is in this condition, in diabetic condition, is very important. So that is why we targeted how this phytochemical camphorol is going to affect the functioning of the autophagy process. So just brief for the students. So you know, autophagy exists in 3 different forms: macroautophagy, chaperone autophagy, and microautophagy. I have focused this, I have marked this as red because this is my major topic for today's discussion. So just for information, so there are 3 types of it. This is macroautophagy. So what happens here? So if you imagine, then the cell, some organelles are damaged, so they have to be destroyed. So first there is a formation of phagophores. Formation starts initially, then it recruits endosomes, and it becomes a complete spherical structure and engulfing the molecules or the cellular organelles to be destroyed. And once the autophagosome, this is called the autophagosome, that is the membranous structure which engulfs all the molecules to be destroyed. And then it brings and fuses with the lysosomes, and then it destroys the whole cellular organelles, damaged cellular organelles, or the protein or any other molecule that has to be destroyed. And then whatever is left out, good ones are given out to the cell in the form of cytoplasm. So this is one sort of autophagy that we talk about in general process. Then we have another one. This is called the chaperone autophagy. What is that? Basically, there are certain proteins which has got KFERQ motifs. These are the amino acids. When these motifs are there in the cell, so in these proteins, so they are actually bound by a chaperone protein which is HSP70. So HSP70 binds to those proteins having these particular motifs and brings it to lysosomes. And in the lysosomes, so there are some membrane receptors, what is called LAMP2A. There are basically receptors which in turn accumulates a oligomerases and takes the protein to be degraded inside the cell. And once they are inside the lysosomes, they are destroyed. So this is one form of chaperone-mediated autophagy process, which also is taking place in the cell for the autophagy. And third one is the microautophagy, which is taking place when there is some damage has to be done very close to lysosomes. They just sequestered out by the lysosomes, and they are destroyed out. So these are 3 out of these 3. We are focusing mainly on the macroautophagic process because that is the process which is mainly involved in the diabetic conditions or obesity-linked conditions. So now, why this autophagy is so important? So autophagy is linked to the, as I told you in my topic here, title here, I said lipotoxic environment. Lipotoxic means when we have the fatty acid. So fatty acid means lipids are there. So free fatty acid is one such fatty acids which actually damages the function of pancreatic beta cells. So if you see here, so this is when any cell, like for example, pancreatic beta cells comes across fatty acids or fatty molecules. When they comes to it, so they starts to degrade the proteins, damages the organs. They starts this process. So if this happens, the cell rescues out for autophagic activity. To survive, they need to undergo a autophagic activity. So if you imagine a pancreatic cell is coming across to a high amount of fatty acids or fats, so what will happen? Those fats will destroy the cellular proteins, mitochondria, etc. And then cell, if they want to survive, they need to undergo autophagic activity. So if the autophagy activity occurs there, so what will happen? It does not occur. Then what will happen? The pancreatic beta cells will die. Cell death will be more. Proliferation will be less. So poor beta cell functions, and this will lead to diabetic condition. But if the cell at this point of time, if it undergoes autophagy, that means it can sequester these damaged mitochondria or damaged proteins. So what will happen? Cells will proliferate faster, death will be less, pancreatic cells will rejuvenate, and that will lead to avoid the diabetic condition. The person can be cured. So that means if we can induce autophagic process in a cell by any means, by drugs, any chemicals, phytochemicals, nutraceuticals, if we follow this pathway, then we can actually rejuvenate the pancreatic beta cells and cure the patients from the diabetic condition. So that was actually our goal in the study. So we wanted to see, does camphorol has any role in inducing autophagy in the damaged pancreatic cell and rejuvenate it? That was our question. So we wanted to address this question. So we went in a step-by-step fashion. So I have made the presentation in that way that students who are who want to be future researchers, so you have to address, when you address such questions, you have to go step by step. You cannot jump directly. So this is why I have shown this presentation, made the presentation like that. So when you see, this is the first step was that how does camphorol really affects the viability of these pancreatic beta cells? So for that we took a RIN5F cell. So what is this? This is basically a rat islet cell line. So generally it's in vitro condition. So we do experiments first in vitro, then in vivo. So we grew these cells. So this is a cell from ATCC. So when the cell grows, in the in vitro condition, it looks like this. So this is the in vitro condition cells. And when they proliferates, they makes multiple copies. So the cell number increases. So this is how the cell looks when they are grown for a few days. This is called high density cells. So this is our model for the cells. So we took the cells, grew these cells, and to make the fatty conditions, we treated these cells with high fats, like palmitic acid was our fats here, and then we treated the same cells with camphorol, our test chemical or test drugs here. So the first experiment was that, so does our drug or camphorol induces the protect the cell from the dying out, or they can keep the cells intact? So for this we do one very important assay called MTT assay. This is basically a cell viability assay. So now you can see when we treated this RIN5 cells with increasing concentration of camphorol, it did not do any toxic effects, so that it induced the proliferation of these cells. So this was an indication, okay, that means the camphorol has cell proliferating properties. So this was done by MTT assay, and this is another assay called BRDO assay, where we see the incorporation of thymidine, synthetic thymidine in the cell. So thymidine incorporation means more of cell proliferation. So what we could see, that camphorol was not at all toxic to the cell, rather it was cell proliferating. It was rejuvenating the pancreatic beta cells. These are the normal conditions. So now once we have confirmed this one, then we move to say, okay, what happens to these pancreatic cells if we induce them with the fat or palmitic acid? So palmitic acid here is a representative of fat. It is basically a fat inducing molecule. Now you see the interesting thing here. So this is our RIN5 cells. When I treat these RIN5 cells with palmitic acid or fat, the red dots indicates the decomposition of fat. This is oil red staining. Red dots indicates the accumulation of fat in the cell. So that means if you induce, so that means if the pancreatic cells are exposed to fats, so the cells become fatty cells. These are fatty cells. Now what happens? So this is normal cells. When I added camphorol, no fat deposition, nothing is there. Now what is interesting here is there. So if I treat the cells with palmitic acid and then I treat with camphorol, so what camphorol does? You can see the lipid amount is reduced. The amount of fat in the cell is reduced. So that is shown over here. So when I treat the cells with palmitic acid, fat amount of the cell is very high. Now we treat them with camphorol, you see the fat amount drops down. So what happens to this fat? That means camphorol actually dissolves the fats out. So it can rescue the cells, that the cell become modified. They can remove the fats that is formed within the cell. So we check for triglyceride concentration in the cell, same thing, high amount in the presence of palmitic acid. But if you treat them with with this camphorol, this drops down. Now we want to test. So what I am saying, is it true in the cells? So what we do, a body P is basically a fluorescent dye, which actually stains specifically the fat cells, in the fats in the cell. So you see in the control cell, no treatment was there. Cells are, no green coloration is there. We treat with camphorol, no fats, nothing is there. But if I treat the cells with palmitic acids, so you see the green deposition occurs. So green color indicates the palmitic acid got deposited in the cell. That's why body P was staining the cell. Cells are all green. Now, if I treat this with camphorol, 10 micromolar concentration, you see the fat portions are gone. So fat portion is no more there. So camphorol was removing the fat from the cell by some means. So earlier you saw it was inducing proliferation. Now you saw that it was removing the fat from the cell. So even if you are obese in a condition, so if your body is having large amount of fats, so if it deposits in the pancreatic cells, but if you are consuming large amount of camphorol, so probably you can overcome the fat deposition in the pancreatic cells. So this was a very important clue that gave us, okay, really something was happening. So camphorol was doing something in the cell. So when you went to forward further, so now what we wanted to see in the next phase was, like, camphorol was, the palmitic acid was inducing fat in the cell. So this fat induction is it linked to something? Is it causing any damage to the cell, or just deposition of fat? So now we see when you treated the cells with palmitic acid, fats, you see the cell viability was reduced. That means fat deposition was not only depositing there, but was killing the pancreatic beta cells. Now, what is the way out? So if I treat these same cells with camphorol, you see the cell proliferation is again rescued out. So the cells who are dying, they are rescuing out. So that means palmitic acid induced toxicity was getting rescued by the camphorol cells, camphorol in the cells. So camphorol was preventing the death of the pancreatic beta cells. So this we showed by different ways. This is by cell viability assay. Now you can see this is normal pancreatic beta cells. When you treat them with the palmitic acids, you see the cell, all this, you can see dotted structures are there. Dotted indicates the cells are undergoing apoptosis. And now, if I treat them with palmitic acid plus camphorol, you see the dotted structures are gone. That means apoptosis is inhibited. So with higher concentration, they are all gone. So the cells are rescued. These are all pancreatic beta cells, and the nucleus, these are nuclear stains. So this nucleus of the cell is deep blue, which indicates the nucleus are very healthy. Now, you all know that apoptosis is a process which is marked by the DNA ladder formation. So here you see, when we took these cells, treated different categories of treated cells, we took the DNA out of the cells and ran a gel. You can see this is a palmitic acid treated cells. You can see the ladders are there. So ladder indicates the cells are undergoing apoptosis and they are dying out. But once we start treating the same cells with camphorol 1, 10, and 50 micromolar C, then ladder is gone. Ladder is gone means the cells, there is no apoptosis. The cells are there surviving. So that means camphorol was protecting the cells from the palmitic acid induced toxicity or lipotoxicity. So this was a very important clue that we got from here. Now the question was to address is to see what is happening at the genetic level. Does the genes responsible for apoptosis are also prevented by camphorol? So you see in this panel, we are trying to show a large number of genes, caspase 3, BAD, BAGS, BCL2, BCLXL, that are the genes responsible for apoptosis. So if the genes are expressed, so there will be apoptotic process. So now when you check for the genes in these cells in response to palmitic acid, you can see they are all elevated out. That means the cell is undergoing apoptosis or cell death. Now what happens if I treat these cells with camphorol? So when I treat the cells with camphorol, you see the level of these genes which are getting upregulated are much reduced. So all the proteins or the genes expressions were significantly downregulated. Now, if you might say this might be an artifact. So to test that, we use temsirolimus, which is basically a known autophagy inducer. So this induces autophagy, positive control drug. So you can see when you treated palmitic acid plus this positive control drug, this is a way of doing an experiment. When you do an experiment, you have to show it as negative control as well as positive control. So this has to, I have to establish that. So you see when I added temsirolimus, you see it was inducing autophagy. So if autophagy is induced, so all the gene expression was reduced down. And now in the same platform, when I treated my cells, these cells with my camphorol, you see it was saying the same response as the drug. Sometimes it was better than the known drug. So that means our drug was really acting and inhibiting the formation of the apoptotic process by downregulating all the genes responsible for causing cell death or apoptosis in the cells. So this is basically a cleaved caspase 3. You know, caspase 3 is one of the important protein responsible for cell death or cell destruction. So when I treated the cells with palmitic acid, very high level of caspase 3, then when I add temsirolimus, the positive control drug, you see it is reduced down. And when I treated with my drugs, there is camphorol, you see that the cell, this caspase level is much more reduced down, significantly reduced down. That means camphorol was inhibiting the apoptotic cell death. So this is how we could establish that this palmitic acid was inducing fat was removed, and the apoptosis of death, cell death was rescued by the camphorol in a dose-dependent manner, and it was same as that of the positive control drug, which is used as an autophagic inducer. So now we wanted to establish that was it really autophagy now? So we know this palmitic camphorol was relieving the cell or rescuing the cell from the cell death. But how to link the use of autophagic process? We have already shown one clue there because I used a positive control drug. Now we have to establish the mechanism behind it. What was the mechanism? So if you see the autophagic process, so autophagy involves different stages, as I showed in my previous slide. First, there is the formation of membrane. So this is the membrane, means it will start to surround the cells or the organelles or the proteins to be destroyed. So it will form the autophagosome formation will start. and then this membrane will become complete, and then it will form a autophagosome structure surrounding the cells or organelles to be or cell organelles or the proteins to be destroyed. And then this whole structure will get fused with the lysosome, and then after the fusion, all this structure, all this cellular, whatever has been brought by the autophagosome will be destroyed, and the cell gets free of those damaged or destroyed structures. So for this, there are different players in this process. So you can see a large number of proteins are involved. So now you cannot target all of them. So we selected whatever circled here, we selected those proteins to check how they are doing it. Are they real? So if I actually selected some of these proteins here, I selected some of the proteins here, I selected some of the proteins here, and in all the steps I selected different proteins. So to check at which stage camphorol is working, at the initial stage, at the middle stage, at the advanced stage, or at the late stage. Now let me tell you one very important thing. I will be using my next few slides, this MDC. MDC is basically a marker for autophagosome, means till the formation of this. And lysotracker indicates it actually stains the lysosomes. So if this occurs, so that means when the autophagosome fuses with the lysosome, so you will see the staining of both MDC and lysotracker. Otherwise, if there is only this process is going on but no lysosome formation, you will see the staining of only MDC. And if only this is happening, not this one happening, you will see the staining of this lysotracker. So we have used these two different dyes in our future experiments to show that what is happening at which level the camphorol is acting. And these are the different marker proteins which we have used for our study. So to show that these are the proteins involved. So if I see here, like ATG7 is a very important protein responsible for forming the autophagosomes. LC3, LC32, etc. they forms the autophagosome formation. So responsible for that. And mTOR and AMPK is actually the protein responsible for initiation of autophagy. So they will start the autophagic process. So this is how the whole steps goes in. So now, so these are what we are looking for. That we are seeing that pymidic acid actually induces mTOR activities, and it inhibits the fusion of the lysosome with the autophagosomes. So naturally, this autophagy is inhibited. While if I see the camphorol function, camphorol is actually stimulator of AMPK, and that's how it induces the autophagic process. So we'll see it experimentally how this is happening. So now this is what you see here. So when I treat the cells with DSA control, nothing is there. So no early stage markers, no autophagosome, no lysosomes, nothing is there. When I treat the cells with say camphorol, so camphorol induces autophagy in the cells. So you can see the expression of the initial autophagosome formation is there. Lysosomes markers are there, and when you fuse them, so you can see an intense coloration there. Temsirolimus, as I told you earlier, autophagy inducers, it induces autophagy in a cell. You see this MIDC trackers are there, autophagosome formation is there, lysosome formation is there, and intense strain. What happens in the pymidic acid conditions? If I treat the high fat condition, you see, so this initial autophagosome formation is not there, nothing is there. It inhibits lysosome formation. So naturally, what will happen? The cell accumulates a large amount of fats, accumulation of large amount of damaged proteins, mitochondrial organelles, etc. So they are all accumulated in the cell. So the cell is about to die. So now what happens if I treat the same cells with camphorol? You see, it rescues back. So autophagosome formation starts, lysotracker formation starts, and when you overlap them, so you can see they have all divided back. So that's how camphorol protects, by inducing the autophagy in the cell, and thus protecting the cell from further damage and inducing the autophagic process, which was done by the pymidic acid. Now, if you see here, this is how the whole process is. So they say, for example, mitochondria protein peroxisome has to be damaged. It has to be damaged. They have to be removed from the cell. So first, there is a formation of membrane formation starts here, and then during this process, LC3-2B recruitment starts. So this is the autophagosome formation engulfing the organelles or the proteins to be damaged or degraded. During this process, LC3B protein recruits another protein called P62. Now mind it here. So once this is produced here, so after some time, these autophagosomes will fuse with the lysosomes, and then they'll form a autophagosome lysosome, and this is a complex called autolysosome. So fusion of lysosome with this autophagosome, and once this fusion occurs, so this is the destruction of the cellular organelles, proteins occurs. During this process, two important things happens. The concentration of LC3B, what I will call as LC32, goes up, and P62 protein, which was recruited from here, their level goes down. So their level from here, their level goes down. So if you find in any case situation, P62 protein level going down and LC3B level going up, that will indicate your autophagy process is complete. That's what our goal was. So we took these two markers, and that was our marker. What was the status of P62 in a cell, and what is the status of LC3B or LC32 in a cell? That is what we will see here. Just mind it. P62 level will go down because it is getting destroyed after this process of autophagosome formation, while LC3B remains in the cell in a high concentration. That was the whole clue here. So now, before I move here, so this entire step, what happened, how the recruits. So this is the LC31, and LC31 in turn, phosphatidylamiine is another biomolecule that is recruited by LC31 and forms a complex with P62. And this is the protein which is to be ubiquitinated and brought to the autophagosomes. And then after this, so you can see here a complex of P62 protein to be broken down or ubiquitinated, and then this LC32 complex is there. So this structure is called LC3 puncta. We call it as LC3 puncta in the autophagic study. So now we are trying to see it. What if any cell, if you see the formation of LC3 puncta, you have to imagine that autophagosome formation has taken place. So this is how we look into it. So how we see it here. So you can see this is a cell, and what we did in our experiment, we transferred the cells with EGAP LC3. That means we transferred this construct, LC3 gene construct with EGAP, means green fluorescence protein, to this cell, pancreatic beta cell. What will happen? So you see the initial cytoplasm is entirely green because all the LC3 proteins are in the cytoplasm. Now I induce a cell with autophagy. So I induction of autophagy starts. So what will happen in the autophagy induction starts? So then there is the formation of this autophagic autophagosome starts to form. And if it starts to form, what will happen? There is upregulation of these proteins, ATG7, ATG3, they are responsible for autophagosome formations. So you see all the cytoplasmic LC31 goes in and accumulates in the autophagosomes. So now see the color of the cytoplasm is now no more that green as you saw here or here, because all the green color was because of LC3 protein. Now LC3 has got accumulated in the autophagosomes, and they are now here, not in the cytoplasm. Cytoplasm becomes pale now. So now this is, if you look at this structure, so these are basically the autophagosomes. So if any cell have this structure formation, we see that autophagy process is going on in these cells. So this is called the puncta formations. So now you see the same thing what we did in our cells. When we treat the cells with BSA, since it does not induce an autophagy, whole cytoplasm of the cell is green. But if you treat the same cells with camphorol, you see the cytoplasm becomes very pale like this, and there's a dot-like structures indicating formation of autophagosome. So that means camphorol induces autophagosome formation. How to prove this? So use again temsorol mass or positive control. You see the same thing what with camphorol. But what happens if you treat the cells with palmitic acid? You see it inhibits puncta formation. So cytoplasm is again green as like this. And when you treat with camphorol, you see it rescues somewhat. So some of the puncta formation occurs, and otherwise the background of this green background of control or palmitic acid condition is gone, indicating camphorol rescues the palmitic acid cells by formation of the autophagosome. So that was actually showed that autophagy is induced by the camphorol. So now I have to establish this fact experimentally also, as I just now told you. If P62 concentration goes down, that indicates autophagosome formation is taking place, and so that is occurring, and LC3 amount should go up. So same thing happened. We treated the cells, pancreatic beta cells, with camphorol. You see the expression of this protein P62 came down, and LC32 expression went up. And this is with the increasing concentration of camphorol. While in the palmitic acid, you see the expression of P62 was very high because it inhibited autophagy formation, and also it inhibited the LC32 production. But if you treat the same cells with camphorol, the P62 concentration gone down and LC2 is up. So that showed that our drug or camphorol was working to protect the cells. So in a time-dependent manner, same thing happened. So when you saw the cells, in this case also, LC2 concentration increased in a time-dependent fashion, while P62 concentration went down. So that means camphorol was really causing the autophagy process in the cell. Now autophagy inhibitors. So now I have to show this by inhibitors of autophagy that what camphorol was doing. Is it really the fact, or it is some artifact? So I have to use some inhibitors of autophagy. So I have used stimulators. Now I have to use the inhibitors. So what I did, I took different inhibitors there. So what menin is an initial stage of autophagy inhibitor. Then we took ATG7 siRNA, which are the middle stage autophagy inhibitors, and then we use chloroquine. This is the same hydroxychloroquine which is now being used for the COVID treatment also, which actually inhibits the lysosomal formation. So that means if any of this is added, so they will stop the autophagy formation at different stages. What meaning as initial stage, ATG7 at middle stage, and chloroquine at the last stage of lysosome formation. So all these are inhibitors of autophagic process. So it will inhibit the phagophore formation. This chloroquine will form the autophagosome formation. That means fusion of this lysosome, and this will be inhibited. So now we use this as markers in our system, and so these are the next few data will show like this. So you see here, so when you treat the cells with palmitic acid, it inhibits both MDC and lysotracal, means initial stage autophagy is inhibited, lysotracal is inhibited, so lipids is inhibited, so all this is inhibited. But if you treat the cells with camphorol, it induces the initial stage autophagy formation, it induces lysosome formation, so the staining is bad. But what happens if I did it with Wortmannin, which inhibits the initial stage of autophagy? So it is not there. Since initial stage autophagy is inhibited, lysosome inhibition is also there. And what happens if you treat the same with camphorol? You see the initial stage was not there because Wortmannin inhibited it, but camphorol induces the lysosome formation. And then what happens if you treat the cells with chloroquine? You saw it was inhibiting lysosomes. So there was still some expression of the initial stage of autophagy, but let us say lysosome is not there. But when you treat the camphorol, so camphorol induces the production of initial stage, but since chloroquine was there, so it stopped the lysosomal action. So even camphorol could not protect it out. So it was not there. So it was only the first stage was there, second stage was not there. That means this experiment actually showed that camphorol actually was involving the entire stage from the initial stage. So if you block any one of the stage at the initial stage with Wortmannin or late stage by chloroquine, your autophagic system is blocked. So even camphorol cannot protect the cells as compared to what it did independently. So autophagy was really involved in this process. So we showed by routine experiments involving the inhibitors here. So you can see if we treat the cells with camphorol, P62 level low as expected, LC3 too high. Now if I block this with Wortmannin here, you can see with Wortmannin. So if I had the Wortmannin here, P62 is very high, LC3 level is low. But if you treat them with camphorol, it rescues to some extent. So that was the major finding it here. Same thing when it blocked with ATG7 with siRNA, same thing. So if in the normal condition camphorol protect the cell, so P62 level is low as compared to this one, and this LC3 level is high as compared to this one. But if I block them with AT7 siRNA is an inhibitor. So you can see here the P62 level goes up and LC3 level goes down. Now it is partially rescued by when I add camphorol, it rescues partly of that. So P62 level is a little bit lesser than this one and LC3 level higher, but it cannot completely rescue because AT7 is very critical for the formation of autophagosome. So we showed this by using the LC3 expression by immunofluorescence analysis. So this is what LC3-2, as you can see here, is when we treat them with the palmitic acid and camphorol. So high level LC3 expressions, but as soon as we treat them with Wortmannin, this is gone because it inhibits the initial stage of LC3 production. So camphorol cannot protect it out. And if you use chloroquine is a later stage inhibitor. So you see there are some expression of this LC3-2, and if you use camphorol, this goes up. But since it is an initial late stage one, so it does not affect the initial stage, but it affects the later stage. So now, so what does it involve? What pathway is involved in this process? So we now next wanted to see the AMPK and mTOR pathways are responsible for autophagic process. So this is camphorol. It stimulates AMPK. And so how much time is left for me? Can you please tell me? Sangeeta?
So you continue, sir.
Is it okay?
Yeah.
Okay. So this is camphorol, and camphorol actually stimulates AMPK. It's adenosine monophosphate kinase, and it induces autophagy. And there's one more molecule called mTOR. mTOR actually is inhibitor of autophagy. So this AMPK and mTORs have inverse relationship. They are enemy to each other. So if AMPK is high, so it will induce autophagy. If mTOR is high, it will inhibit autophagy. So we wanted to see what this our camphorol is doing. Does it doing AMPK activation or inhibiting mTOR or activating AMPK and mTOR both simultaneously or mTOR? So this was what we wanted to see in our next experiment. So what we did, so you see this is a expression of mTOR in a cell, and then with time dependent way when you saw it, you see with time the expression of mTOR went down, and while the expression of AMPK was upregulated. So as you treat the cells with camphorol, at 72 hours, you saw the minimum amount of mTOR and maximum amount of AMPK. And this was a time-dependent form. So now we use different concentration of camphorol. Now you can see here. So with increasing concentration of camphorol, so when I see the cells with palmitic acid, you see palmitic acid induces the production of mTOR, because palmitic acid inhibits the autophagy process. So it induces the production of mTOR, and it inhibits AMPK. So now what happens if I treat the cells with temostral autophagy inducer? You see, it reverses the thing. So it inhibits mTOR and upregulates AMPK. Now what our camphorol is doing? So when you see it out, you see it immediately inhibits the mTOR production. Biosynthesis of inhibited. At the same time, it was upregulating the AMPK activation. So now this was proved that camphorol induces the production of AMPK, phosphorylated AMPK, and inhibits the activation of mTOR. So it was doing both of them simultaneously. So it still remains a question: is it doing only? So that means the downregulation of mTOR is it because AMPK is not there? Because in my previous slide I showed you AMPK inhibits mTOR. So does camphorol doing only AMPK inhibition and then subsequently mTOR, or it is upregulating AMPK at the same time inhibiting mTOR? That was the second question. So this shows that, okay, this clear cut shows that AMP is upregulated, mTOR is downregulated. Now to address the second question of what I just now asked you, so we wanted to see what happens if I inhibit AMP. So compounds is an chemical which inhibits the AMP. So what happens if the AMP is inhibited? So now you can see in this situation here. So this is palmitic acid, mTOR is there, and so camphorol is not there. So AMPK is less. Now if I treat the cells with camphorol, the level of mTOR is less, AMPK level is very high. So this is what the situation is. So now this is a good suggestion, is that mTOR level is completely gone. In 10 micromolar concentration of camphorol, mTOR is gone, AMPK is very high. Okay, this is what we expect. LC32 is high, P62 is low. This is the most ideal situation what we have. Now what happens if I treat the cells with compound C? CC indicates compound C, which is a basically inhibitor of AMP. So what will happen? So you see immediately the AMPK level drops down, because it's an inhibitor, it inhibits it. So if it is inhibited, naturally mTOR will be upregulated, because you know AMP and mTOR are enemies to each other. So if one enemy is gone down, other enemy comes up. So mTOR comes out. So this is mTOR's level is very high here. So now our question was, so if what happens to this situation if I add camphorol? Now you see when I add camphorol, so AMPK is not here. So obviously AMPK is down already, because we have already used the compound. So AMP is out of question. So now interestingly what you see here, the mTOR is also not there. So AMP is not there. Still the level of mTOR was very low. So who made it low? That means this was camphorol. So that means this shows that camphorol was not only upregulating AMPK, but it was directly inhibiting the mTOR expression also. So that is what a very important clue we got from this study, that okay, this is an important pair. So camphorol was playing at both the levels, inhibiting mTOR, because otherwise at this point of time we should have expected the level of mTOR as good as this one. But because you have already have the chloro compound C, which is inhibiting the AMP, so AMP is not there. So whatever inhibition that you see in mTOR is because of camphorol. So that is what is a very important clue for us, that camphorol was inhibiting AMPK as well as mTOR. So this same thing we checked using an siRNA for AMPK. Same phenomenon we could see, that when you blocked the AMPK, so this has blocked AMPK. Now when you add it with this siRNA, the AMPK level went up, and you see there was some inhibition of mTOR also, as mTOR was absent here. But if you treat with camphorol, mTOR level goes up. So mTOR goes down. So that means mTOR was inhibited both by camphorol here in this process. So it was upregulating AMPK, it was downregulating mTOR process. So this was also already shown by our study also, like when you see the LC expressions in a cell. So this is the camphorol treatment, LC level is very high, and if you treat them with the compound C, you see LC2 level goes down, because it inhibits the AMPK activity. So AMPK is not there, so naturally this does not happen. But when you add camphorol, it partially rescues. You can see some green cells, so LC expression. So it is trying to rescue the cell from the no autophagic condition. So that is how we could find that this part, that we could see mTOR was really involved in this process. Now, the very important question that came to our mind, okay, everything was happening, autophagy was happening, this camphorol was protecting it. So does it have any role, or just autophagy was inhibited? So what autophagy was doing in the cell? So to address this question, we went back to the very basic and fundamental question addressed in this question. So we took the cells and performed a simple cell proliferation assay where we used all the inhibitors, early stage, what mean in early stage. Compound C is basically an AMPK inhibitor. Chloroquine is basically lysosomal inhibitors. We use all the three inhibitors, and you can see in presence of this camphorol, the cell proliferation was increased. But when you treat the cells with palmitic acid, the cell proliferation was decreased. This was this we have seen in my first few slides. I've already established this. Now, if I treat them with palmitic acid and the camphorol, it is rescues out. This is what we know it. But as soon as we block this autophagy process by warfarin, you see the cell viability is gone. Cell is dying out. So that means autophagy is required. So if you block the autophagic pathway, camphorol cannot rescue it completely. This is one. Secondly, if you add chloroquine, which was basically the lysosomal inhibitor, same thing, the palmitic acid cell death from camphorol rescues it out. But if you add here these inhibitors, no cell rescue, cells are dying out. So a second thing of compound C, which is basically an inhibitor of AMPK. So AMPK, so same thing, palmitic acid cell death, this camphorol rescues it out. But if you add compound C here in these two processes, cell death is occurring. So AMPK, if you block AMPK, early stage autophagy, late stage autophagy, whatever you block, camphorol cannot rescue the cells. That means camphorol needs the activation of the autophagic process and AMPK activation to regulate the cells or protect the cells from the death by the palmitic acid induced conditions. Same thing with the DNA ladder. So you can see palmitic acid causing DNA ladder formation. If you add camphorol, DNA ladder is gone. That means cells are all rescued. But if I keep on adding inhibitors of autophagy by one, what mean chloroquine and compound C, you see all the ladders are coming back. So camphorol cannot protect the cells anymore. If you stop autophagy, the activity of camphorol is gone. That establishes that camphorol is really playing an important role in the process of autophagy in these experiments. So now we wanted to establish this one with animals. So I show last few slides where we induce the same thing in the animal model. Whatever I have shown you in the in vitro, now we want to in vitro. So we fed the animals with a high fat diet. So this is an obese condition, and this is basically an obese animal. So you can see the normal mouse. And if we treat the animals with a high fat diet, so the animals become obese. And now if I treat them with camphorol, you see these animals are treated with different conditions. So this chloroquine actually is basically an AMPK inhibitor. So you see the animal is still obese. If I see the weight of the animals, it's as good high fat diet and chloroquine treatment because it inhibits the AMPK activity. So the animal is obese. But if I have high fat diet but treated with camphorol, animal is thin. So it is much thinner and thin. So if I have high fat diet, camphorol plus chloroquine treatment, it rescues to some extent but not complete. It's better than this one. And this metformin is a well-known drug for diabetic condition. So you see it's normal and as good as our camphorol. So that means in the in vivo condition also this was rescuing. So now how to show this? So we have to do several experiments, very simple experiments that we do: fasting blood glucose level, glucose tolerance test, and insulin tolerance test. You can see here when we have high fat diet in animals, so blood glucose level is very high. But if you treat the animals with camphorol, you see this level drops down. So blood glucose level is dropped down. So it is improving the diabetic condition. But same thing with glucose tolerance test. That is the normal test that we do for diabetic conditions. You see in the high fat diet animals, the glucose tolerance is very high. But if you treat them with camphorol, which is shown over here, this purple color, you see it's all as good as the normal animals. So it brings down the glucose tolerance also. At the same time, it brings down the insulin tolerance to a normalcy also as compared to the high fat diet conditions. So same thing when we stop this in this case. So this is the insulin concentration. So you see high fat diet level of plasma insulin is very low, but camphorol treated animals, the insulin level is high in blood as well as in the pancreatic. So we took the pancreatic tissues out, extracted the insulin and estimated it out. It is high as compared to the high fat diet animals. And so this is the plasma insulin after glucose administration. So you can see it is rescuing so if the glucose after food intake, so it was rescuing the animals. So these are the control animals, and you can see purple one, it is as good as the control one after the insulin level was normal even after glucose test. So animal was just normal even after the food intake. This is a very important slide. So control animals. So if the animals are fed with high fat diet, pancreatic tissues are damaged here. And if I treat the animals with chloroquine, as you all know, is basically an AMP inhibitor. So it inhibits autophagy. You see all the pancreatic beta cells are damaged. And if you treat the animals with a high fat diet plus camphorol, you see the pancreatic tissues are rejuvenated out. So it is almost not same, but it's almost same to this one. and it's functional pancreatic. I showed you earlier, it is releasing insulin and very healthy conditions. And if you have chloroquine there with along camphorol, it rescues only partially. Next question was, does this cell are able to secrete insulin as good as normal cells? We performed an immunohistochemistry. This actually stents the in situ insulin content in the cell. So this is a high-fat diet, strep animals. So you see the fragmented cells. So insulin secretion is very less. But if I treat the animals with camphorol, you see the intense coloration of red color indicates the fluorescence shown by the insulin. So high rate of insulin in these pancreatic beta cells. And in case of chloroquine, where AMPK is inhibited, you see the pancreatic cells are all damaged. Insulin level is very low. If I treat the high-fat diet with chloroquine-treated animals with camphorol, it rescues partially, but none of them is as good as this one. So pancreatic cells are rejuvenated, not only in terms of structure, but also in terms of function. So then we do an electron microscopy study to show the autophagosomes formations. So, in the autophagosome can be seen on a microscope. So in the high-fat diet, there's no autophagosomes. But we have the animals treated with camphorol. You see these black dots indicates the formation of autophagosomes. So that means these are the autophagosome formation, and they are the main responsible for rescuing the cells from dying out and protecting the cells from diabetic condition. If you have chloroquine, it inhibits AMPK action. Naturally, there is no autophagosome, only few autophagosome formation. But if you treat the same animal with camphorol, you see some of the autophagosome formation comes out, but none of them is as good as camphorol treatment. So this shows that this is a very important process, and it involves at all the levels. So next we check for the genetic level. We isolated the pancreatic tissues and saw for the expression of this. So you can see high-fat diet, P62 level is very high. As I told you, LC3 level is very low. But in the animals treated with camphorol, just reverts. P62 level drops down and LC3 level goes up. And you can see AMPK level is also very high. So this shows that not only in the in vitro cellular system, same thing was applicable in the animal models also, which you could rescue. So when we added chloroquine to animals, same thing. I just showed you that P62 level is down, and so when we block with chloroquine, this process is inhibited to a great extent. So again, the same thing, insulin functions. When you block this with chloroquine, you can see with the chloroquine, plasma insulin level drops down as good as high-fat diet, but with streptozotocin, this is very high. Camphorol is very high, and in case of this camphorol with chloroquine, is partially rescues out. So it can improve all the condition, pancreatic insulin content and the glucose tolerance. So it was improved. So, but chloroquine, AMPK, if you block AMPK, all these functions are inhibited. That means AMPK is very critical factor, and camphorol rescues this out, and camphorol utilizes them. So if I summarize the whole thing, what I have done still now, so this shows like this: palmitic acid induces the mTOR, and mTOR is basically palmitic acid induces mTOR, and mTOR inhibits autophagosome formation. This is clear. Palmitic acid induces apoptosis of the cell and thus inhibits the beta cell survival. But autophagy actually is a process which inhibits apoptosis and induces cell survival. Who does it? It is basically by the process of camphorol. What camphorol does? Camphorol upregulates AMPK. AMPK in turn inhibit mTOR. And if the mTOR is inhibited, then whole of this function is all blocked. So that means it's all inhibited activity of mTOR is gone. So if this is not there, so naturally the camphorol can activate the whole process of cell survival, autophagy, and rescuing of the cell. That is how our whole process continues, and camphorol plays a very critical role in this process. So in conclusion is that camphorol exerts a cytoprotective effect on palmitic acid-induced pancreatic beta cells by induction of autophagy. AMPK-mTOR signaling pathway is involved in camphorol-induced autophagy, and camphorol could be used as a potential nutraceutical candidate for prevention of obesity-linked type 2 diabetes. Because you have to understand this nutraceutical market is extremely high, and it is expected it will grow about 7.6% by 2025. So it's a very important molecule, and it's very important that we should work in this particular direction to give scientific validation to this one. So not only extracts, not only like that, you have to give a scientific outlook to this aspect. This is my whole lab. So Ritu is actually whose work I presented in this one. Ritu finished her PhD recently. So Ritu and others are all in different projects in my lab. So so this is how the whole of my past students who have worked with me still now getting their PhD degrees. And these are my collaborators at different institutes who have helped me in different chemicals, different forms. And these are my funding agencies who have helped me out in different ways. So I come from a state called Uttarakhand, which is a state of hills, rivers, and lakes. Very beautiful scenic beauty. So you are all welcome to visit this state. and especially thanks to you all. And this is our main building, and I have to just inform you one important thing here. This is the James Thomson building, and you know, this is the oldest technical institute in Asia. It is a 180 years old institute, and this building stands 180 years old. Building that is standing here. This is our main administrative building. So this is mainly administrative block. Our departments are different. So that is what. With this note, I end my talk here. So thank you very much for all your patience and bearing me this almost 1 hour now. Thank you so much. Thank you. Sangeeta, I am all in.
Thank you, Dr. Roy. Thank you for your nice and lucid presentation, which is very interesting and informative. Now I request the participants to send their queries, if any, to Dr. Roy in the chat box, or may directly ask the speaker by unmuting their microphone. The session is now open for interaction with the speaker. Mona Lisa, please check whether there is any question in the chat box.
If somebody can convey the questions from the chat box. I cannot see the chat,
Yeah.
see the chat.
Thank you, sir, for your nice presentation. We have 4 questions in the chat box.
Yeah.
The first question is from Dr. Sandip Chakraborty. He asked whether the camphorol can downregulate the ceramide-induced beta cell apoptosis.
Yeah, it's actually, it depends to be seen. We didn't see that portion. We have seen mainly the palmitic acid induced because the impact of different chemicals is variable from cell to cell. Okay, so I'm not in a position to comment on this one. So it might be checked actually.
Okay, sir. Thank you. I think he asked another 2 questions which are related on that thing. So
Can you please tell me if I can have any clue? If you can tell me the question,
okay.
I can have.
Okay, sir. The second question from him is, the camphorol have any role in inhibition of ceramide synthase?
Okay, it is again the same all linked questions. So I'm saying the same thing. Yeah, I think probably need to be seen. What we have seen is basically exclusively autophagic process and in response to lipotoxic environment. So basically, it might vary. I'm not commenting on this one.
Okay, sir. Then I think we can skip the third one also. That is also related. And we have one question from, I think she's one of the students who joined us, and she asked that if diabetes mellitus is a genetic disease.
It is somewhat. There are certain genes which have been identified, which may be linked to the diabetic conditions. So nowadays things are moving in a different direction. So even people talks about the microRNA expressions, even at the epigenetic levels. So if those are linked together, I would say yes. So there are lots, because you have to understand one important thing. Diabetes is not a single gene disorder. It's a multigenic disorder. So if the multigenic disorder means multiple genes are involved, and how each genes coordinates each other, so that is the most critical part of it. So involvement of meat and other factors would play a critical role in the process. So at this point of time, yes, if all those multifactorial things are coming together, so it could be genetic, but this needs a long deliberations because there are several involvement of SNPs and other factors are there.
Thank you, sir.
Okay, if there is no question in the chat box, then we can move to the next session.
Okay,
Thank
ma'am.
you so much.
Please.
So can I quit this one?
Yes,
No, I have already shared.
you can stay with us.
Yeah. I will stay here, but I have stopped sharing. No.
Okay. You are
Fine,
moving. We are moving to the next session. So this is the end of our first plenary session. Now I would like to welcome Dr. Shen. Yeah.
Sangeeta, can you hear me now?
Yes, I can hear you.
Okay, so I am having a problem with video, streaming. I don't mind to present this one. There are two options. So I can quit and come back, which may take 1 or 2 minutes, or I can go ahead and present it, and at the end I can come back and show my video if it works. I think it's cookies is blocking my video streaming. So it's up to you.
Then
I
you
think,
can quit and again join.
are you sure? Because the connection is good now. It's okay, then I can quit it. It may
Then,
take
then
1.
you may continue, please.
Okay, let's continue. And then once we finish the lecture, then I can come back and say hi to everyone, okay?
Okay.
All right, go ahead.
So,
Thanks.
yeah, so we now welcome Dr. Utpal Sen to deliver his valuable talk. Topic of his lecture is Innate Immunity, a crossroad to hypertensive nephropathy. Here is his career profile. Dr. Sen is currently an associate professor and in the final stage of transitioning to a full professor of the Department of Physiology, University of Louisville, School of Medicine, USA. Dr. Sen is well known for his work on hypertension and diabetes. After completing his master's and PhD from the University of Kolani, he moved to the USA for postdoctoral training. Due to his monumental passion in biomedical science, Dr. Sen was appointed as an assistant professor and then secured a faculty promotion to associate professor. He has received many awards in his academic career. Most notables are a postdoctoral fellowship from the American Heart Association, a fellow of the American Heart Association, a research recognition award from the American Physiological Society. He has published over 80 articles in high-impact journals of his research field, including American Journal of Physiology, Journal of Hypertension, Nitric Oxide, to name a few. He has also trained and currently training several postdoctoral fellows, MS students, and medical residents in his laboratory. Dr. Sen is serving as one of the section editors of Pharmacological Research, academic editor of PLOS ONE, associate editor of Frontiers of Pharmacology, Renal Pharmacology. He is also an editorial board member of American Journal of Physiology, Endocrinology, and Metabolism. In addition, he has served numerous journals as an expert reviewer in the past 15 years. He has served and currently serving as an expert reviewer of grants proposal for many federal and foreign agencies that include the National Institute of Health, Department of Defense, American Diabetes Association, and numerous others. Last but not the least, his research is continuously funded by NIH since 2011. I request Dr. Sen to deliver his talk. Over to you, Dr. Sen.
Can you hear me?
Yes,
Okay.
you are audible.
All right. Thank you, Sangeeta. Thanks to the organizing committee, principal, and others who organized this seminar and gave me a chance to talk about our work. And I feel very fortunate to present this one. And I hope I could be in your college. It looks a beautiful building, but unfortunately, it's so far. But I am feeling that I am in your college. So please bear with me. I try to explain my work as simple as possible so that everybody can understand. If I'm too simple, just knock me. I'll be more than happy to explain more, or at the end you can ask me any question you want. Don't hesitate to ask, even if it is you think very silly question. So first of all, I would like to disclose that I have no financial interest with any commercial entities which otherwise may influence my work. Some of the graphics are web-based just for educational purpose to make you understand what we are doing right now. So to give a little background about my work, I will be talking about immunity. As probably you know that there are two main different types of immunities. One is innate immunity and another is adaptive immunity. Innate immunity is basically what we inherit from our ancestors. For example, we have leukocytes, macrophages, monocytes. Those are army in our body. They defense from the intruders like virus, bacteria, and other pathogens to enter and fight them and then kill them, then protect our body. When this immune system is compromised, or that those innate immunity system doesn't work properly, we get very sick. Like for example, in this COVID pandemic, the macrophages cannot recognize or cannot kill. The other one is adaptive immunity. So adaptive is which we don't inherit, but we develop over the time. For example, if someone having chicken pox in their early age, they develop antibodies which resist further infection in their later life period. Those are adaptive immunities. Today, I'll be talking mostly innate immunities, which we inherit from our ancestors. So when we talk about innate immunities, there are so many receptors and so many systems works together. One of them are toll-like receptors. They are part of immune system. There are many different types of toll-like receptor. I will be talking mostly one particular toll-like receptor, which is 4. So what happens, as I said, there are many pathogens which invade our body, and we also have microbiota in our gut. As you know that we get infection, like say, for example, Escherichia coli or Salmonella, we get sick, but we also have microbiota in our gut from the beginning when we born to our death. And that's the composition of microbiota. There are different phylums, different class, different genus. So as we grow or become older, they shift from one genus to another genus, or like I would say, there is a balance between some of the bacteria to the other. Because our food habit changes, our epigenetics changes, so the bacterial composition shifts from one direction to another directions. When the bad pathogen takes over, then we get very sick. So we'll be talking about the connection between toll-like receptor, how innate immunity respond to our microbiota in the gut when we become sick. Hypertension is, we just listened a beautiful talk by Dr. Roy about diabetes, but I will be talking in hypertension, which is another major problem, major health risk, especially if it is uncontrolled. We know that we can develop stroke, or we can even die from this hypertension if it is uncontrolled. But it is also important to remember that hypertension is linked to organ dysfunction. We may not die, but there might be organ dysfunction or paralysis due to the hypertension. And microbiota in the gut, they to some extent control this hypertension, or other way, hypertension can also influence the gut microbiota composition. I will be talking about those relationship between these two in the latter part I might talk. So what happens, actually, we know that there is a link between hypertension, gut microbiota, and toll-like receptor. They control the immune system, and then they are defense army in our body. But whether there is a crosstalk between all these, hypertension, diabetes, microbiota, and toll-like receptor, and whether that could lead to kidney damage is not very clear. So I'll be talking mostly about dysbiosis. The dysbiosis means when there is a disproportional composition of beneficial and harmful bacteria in our gut, that causes dysbiosis. So the biosis or biome in our body or in our gut is not very orderly fashion. So there are more harmful bacteria than the beneficial one, because we also need the bacteria, because many fibers or the nutrient or diet we take, our body cannot digest. So bacteria helps to digest. Most of them are beneficial, mutual beneficial bacteria, which is called mutualism. They help us to digest all these food nutrients we take. At the same time, they get the shelter, plus they get also nutrients. So it's the beneficial bacteria is always good. So the main objective of my study was to identify a crosstalk between hypertension and diabetes. So I will establish this crosstalk once I finish my presentation. You will realize that there is a very good connection between dysbiosis and hypertension. We also recognize the impact of gut dysbiosis in the kidney disease, how this gut dysbiosis leads to kidney dysfunction. There may be other organ function, but my particular interest is about the kidney. In a kidney failure, you might not die immediately, but it's a chronic disease, and end stage kidney failure means, eventually people will die. We'll also demonstrate the benefit of TLR4 mutation. We know TLR4, we need our body, but what happens if there is overactivation of TLR4? It could cause problem, and I'll be talking about how this TLR4 is related to hypertensive nephropathy. So to give you about our hypothesis, how we came up with this idea, I gave you a little background about this work we have done. So what we are thinking, we thought in our laboratory that maybe in hypertension, there is a gut dysbiosis, bacterial composition difference in our gut, which is giving more harmful bacteria, promoting more harmful bacteria than the beneficial, which disturbing the mucosal barrier of the gut. We know that the gut has selective permeability. Gut epithelial layer has selective permeability. In other words, it doesn't allow everything to enter to the bloodstream. It only allows some vitamins and nutrients to enter to the bloodstream because we need those. So it's a selective barrier. When the barrier integrity damages, meaning there is a damage in the epithelial cells, what happens, a lot of other unexpected materials, meaning other unexpected component in the gut, for example, pathogens or the waste material can enter to the bloodstream and can cause problem. So what we are thinking about is that during dysbiosis, there might be a barrier integrity disruption which leads to gut leakage, and ultimately it causes the translocation of bacterial component to the parts of the body, including kidney. When it goes to kidney, it overactivates TLR4, and we know that TLR4 is a receptor which is expressed in many cells, including leukocytes, monocytes, and we need that one for defense mechanism. But if there is overactivation, it could lead to cell death. We learned from Dr. Roy's lecture that there are different types of cell death, apoptosis, ferroptosis, and other things. Apoptosis, he talked about the good thing about apoptosis. Also, he mentioned that apoptosis can lead to the organ failure or even death. So I'll be talking mostly how this activation of monocyte could lead to cell death or a lot of apoptosis, which ultimately damages the organ, particularly the kidney, and lead to microvascular resistance, because we know that kidney has many microvascules, like including all these glomerulars and then tubules, so many microvessel in the kidney, that if there is a cell death or a resistance, ultimately it could lead to kidney dysfunction and failure. So this was our overall hypothesis. Just keep in mind that I'll be talking about dysbiosis and then leading to gut leakage, bacterial translocation to the kidney and causing kidney failure. So is there a way that if we block this TLR4 or inhibit somehow, can we mitigate or improve some of the deleterious effect that dysbiosis is causing? It's possible. So we'll be talking about how this inhibition or blockage of this TLR4 can have some beneficial effect. Just remember that TLR4 we need for defense, but sometimes overactivation can cause a problem. So we chose an animal model. So my most of the work, all of the work over here I'm presenting today is animal model. I have some cellular model which we have experimented for cellular signaling, but I am not presenting this one. All experimental results I'm going to show you in animal model. So we have animal, we got animals. I'll be talking about TLR4 normal. There is a genetic name it's difficult to say all these name over and over again during presentation, so I simplified it. So just remember that HOG is TLR4 normal, meaning they have normal TLR in their body. And TLR4 mutant mice, which is basically there is TLR4, but they doesn't activate. Means they are mutant, so their activation is prohibited. So this is the strain, genetic strain, and I'll be talking about TLR4 mutant. You just have to remember that TLR4 mutant and TLR4 normal. So normal, it expresses, it activates with the pathogens, but mutant, even though there is TLR4, it doesn't get activated. So it's basically doesn't cause any problem. So we have these two mice. So what we made, as you remember, the Dr. Roy mentioned that we need to control, proper control. So what we did here, these animals are normal, meaning they don't develop hypertension. So what we have to do, we have to give or induce them to have hypertension. So what we did over here, we gave angiotensin. If you remember your physiology class, that angiotensin, renin-angiotensin system cause increase of blood pressure, which is basically hypertension in scientific term. So what we did over here, we infused animal with angiotensin. So we have two groups over here, TLR4 normal and mutant, which is we are giving just saline as a control because we are infusing angiotensin, so we have to have a control, and we are infusing this same group, similar group, two groups with angiotensin over the time. So how we gave them, because if you inject angiotensin at one point, animal might die immediately because it shoots up blood pressure so high. So what we have done over here, we had a mini pump. So the pumps looks like something is electrical battery or something working, but this is not like that. This is very simple but a very elegant method to induce to inject or infuse any drug in the body. So what is had basically a small pump, and it is a osmotic pump. What is it? There is a basically cellular layer over here, and we put our drug to this channel. We put that required amount what we need to put over here, and this whole thing we put under the skin. So what happens when this paper, basically is cellulose semi-permeable paper, or they absorb body fluid. So what it gives pressure to this chamber. Ultimately, it oozes out from the body, and it's a controlled way. So the design such a way that it will release small amount of angiotensin over the time. And will not cause any abrupt effect in the blood pressure. So this one, when you load with angiotensin and put inside blood over the time over 4 weeks, we treated the animal. So basically, we took both normal and mutant animal. They are about 10 to 12 weeks old, and then we treated them with angiotensin plus saline as a control over 4 weeks. So ultimately, after 14 or 16 weeks, when they are 14, 16 weeks age, we take them out, measure their physiological parameters, including blood pressure, kidney function, other. I'm going to show you what we have checked, and then we took their body tissue, including kidney and gut, for molecular and genetic analysis. First, I would like to show that the angiotensin we are giving is actually working, meaning that angiotensin is causing hypertension. How to check that? As you remember that we check hypertension or blood pressure through this arm cuff. Doctor comes and put the arm cuff. The similar mechanism over here, but it's a little different for the animal. So we cannot put them on the arm. So we put in the tail. There's a cuff tail. And these machines are automated. So we put cuff and secure the animal over here, because if they jump, their blood pressure changes. So we have to secure the animal in a chamber so they cannot move. And then you put the cuff over there. This is exactly similar mechanism. And you automatically run the program, and your instrument gives the systolic blood pressure and diastolic blood pressure. So if you remember that systolic is when your heart pumps out of the blood, and goes to the different parts of the body, that is systolic pressure. When blood comes back to the heart, that is diastolic, and usually diastolic pressure is always low. For animal, for the mouse, is basically systolic pressure is somewhere about 120, and the diastolic pressure could be 80 to somewhere 90. Similar range. So we want to make sure that the angiotensin we are giving is actually causing hypertension. And here is the result. So there are 4 groups, as I said. There is saline control and angiotensin. These are normal and these are mutant mice. So as you see over here, over time, after 1 week, it started shooting blood pressure in the normal angiotensin treated animals. So they are hypertensive. Whereas in saline mutant and saline normal, we don't see any increase, because we don't expect it. We are giving saline, so there will be no change of blood pressure. That's what we expected, and we have the similar result. When it comes to the mutant mice with angiotensin, as you see here, we expected a blood pressure to have like this, like the control, the normal angiotensin treated animal, but surprisingly we see lesser in between somewhere. So that tells another story. I could go talk on about that story. I think that is also a secondary hypertension. What we see angiotensin induced hypertension may not be exactly or 100% due to angiotensin, could be something angiotensin is doing which secondarily induce hypertension. For example, if angiotensin kills the cells or induce apoptosis or causing resistance, that could lead to hypertension. So that is secondary effect. I'm not going to talk that about. Just keep in mind there is a hypertension, and there could be an angiotensin 2 induced hypertension in normal animal, is maybe the also secondary hypertension is inducing or contributing to overall blood pressure rise. So definitely our angiotensin is working. It's inducing hypertension both in mutant and normal mice, but we see more in normal than mutant. What could be the other cause? Why it is not in mutant mice? Why it is not like the normal TLR4 conditions? Because we believe that I will show you the data that the TLR, because these TLR4 are mutant, they are not activated, so they are not inducing the secondary cause, probably that is inhibiting to make it up to the level. So I'll give the evidence from our result. So this is a busy slide, and probably it will cause some confusion to some of you to understand. I will make it very simple. So remember, we have Microbiota in gut, and there are so many different phylum, different genus, and class. I'll be talking about 2-3 phylum, class in the bacteria, in the bacterial colony in the gut. So what we did, we took out the animal fishes from the 4 groups. We have here all these mutant and normal mice with hypertension. We took the fecal matter and we sent to the genomic code for next generation sequencing. What it does, basically, it gives the genomic composition of all the bacteria. The animals have all these 4 groups. So this is a phylum level. Just focus on these first 3 over here. We have Firmicutes phylum. We have Bacteroids. Firmicutes are this gray number. So if we think about all the bacteria, whatever they have is like 100%. What frequency these bacteria phylum are having? So meaning that what percentage they have. So out of 100%, Firmicutes how much, Bacteroids how much, Proteobacteria how much. So that makes up overall 100%. So if you look these groups, there is a differences. Some of them are higher, some of them are lower here. Particularly in Firmicutes over here, the grayscale, you see there is a changes, and then the Bacteroids, there is also just for the convenience to understand these bacterial dynamics. There are many Firmicutes within the phylum. For example, Clostridium, they are bad, like Botulus or Titinus. They got they are harmful agents. Whereas most of the bacteroids are beneficial, meaning they mutually exist in our body, in our gut. And there are proteobacteria, which is E. coli, Salmonella. Some of them are really bad, and some of them are very neutral. So particularly look these these firmicutes, which is gray, bacteroids, which is over here brown, deep brown, and then pink is proteobacteria. Mostly these three are changes between the groups. So I'll make it very simple to understand. So from there, we extracted the data, and what we found over here, you look, firmicutes are increasing in TLR4 mutant mice. Even in a mutant with hypertension, if it is more than other bacteroids. So these are good bacteria. Firmicutes, some of them are bad, and proteobacteria, there are some bad bacteria. So as you see over here, that firmicutes is pushing, meaning they are overgrowing. The other bacteria is basically getting cornered. So their number is getting less. The good bacteria is getting less, and the bad bacteria is taking over. So definitely something is happening in the gut. So if we take it a little deeper in the class level, we also took genus level and specific level, but I'm not going further details after this class. I'm just to showing you that there is a changes, pattern changes. So again, you see here these gray firmicutes and Clostridia class, bacteroids class in the deep brown over here, epsilon bacteria in the pink color. These are mostly three things, within the three genus, these three different classes are changing pattern. And just remember, in firmicutes phyla, Clostridia is about 90%. So most of the Firmicutes phyla, the Clostridia bacteria. Similar thing in bacteroids, about 80 to 90% are bacteroidea, which is good. And in proteobacteria, most of them are, epsilon bacteria, which is maybe some of them may be Salmonella and other things. So there is a pattern change. There is a definitely, they are not similar in control and treated animal. So if we simplify this one, you see the Clostridia, which is bad, many of them are bad bacteria, is pushing the good bacteria. So they are overgrowing and they are getting cornered, and particularly in the mutant mice with hypertension. So what does tell? Why if they are high, the bad bacteria are definitely they are doing some harm. What are the harms they are doing in our body? To look into details, which, if you remember that I talked about the gut is selective permeable, meaning it allows only certain molecules to enter in the blood, in the blood, like in nutrients, vitamins, and other components, and it prohibits to enter food particles as well as some pathogens to enter. So who actually protects them? There are many molecules we call tight junction protein. So in between cells, there are two cells together. In between cells, there are tight junction protein, meaning they are keeping like it's like a concrete between the bricks. So if you don't put concrete or don't put mortar in between bricks, they are loose. So water can flow through this. But if the concrete is tight, it doesn't allow water. So similar mechanism over here in these epithelial cells. There are so many molecules, tight junction molecules called Jonah, occludin, claudin, and occludin. So we checked whether there is a differences in expression of these molecules. If there is a expression level, means some might be damaged, so they are not showing over here. So what we did here, we immunostate. So we took the gut from all these 4 animals and we stained them with Jonah occludin. You see here in TLR4 normal mice, and then with angiotensin we see less expression, meaning probably these are damaged, so they are not showing at all. And these are the marks. So DAPI is just to show that there is a healthy cells or healthy tissue in the gut, and then Jonah occludin, which is like pink, and these are the marks. So when we took cell or tissues from the TLR4 mutant mice without angiotensin, we see we were expecting that TLR for mutant mice with angiotensin or with hypertension could have been like similar way, but we see no, there are good tight junction proteins, zonooccludins. So that tells us that probably TLR for mutation is protecting the gut. So we have to check other molecules, other tight junction molecules. Among them are claudin and acrodin. So we get similar results. You see, in normal mice, they are expressing normally. In TLR for neutral with hypertension is less expression. That means they're probably damaged. Both are. And with mutant, they are pretty much close to, not exactly, but pretty much close to similar expression like normal. So that tells us that mutant mice are somehow protected to hypertension and preserving their gut integrity. So if the gut integrity is disrupted, what do you expect? You expect something abnormal happening in the gut. So what is these things happening? So what we did over here, let me show you this on the first. So what is happening here? We wanted to check whether the bacterial components translocate to the organ. As I said, like this, our particular interest was kidney. So we wanted to check whether the bacteria translocate to the organ through the bloodstream, because the gut is now disrupted. It is allowing bacterial component to enter into the bloodstream, and once it goes to bloodstream, it can go anywhere and everywhere. So what we did over here, we took kidney tissue and we did 16S ribosomal RNA identification. So as you remember that 16S RNA is only present in the bacteria. So what we have seen over here, when you analyze those tissue, we see in normal condition hypertension, there are plenty of 16S RNA, which is the bacterial component over here. But in mutant mice, these are protected, whether it is like saline control or it's hypertension, it is protected. So that tells us these animals, these mutant animals are protecting the gut integrity and not allowing bacterial components to translocate, whereas these normal animals with hypertension, they are prone to have disruption in their gut integrity and allowing bacterial components to enter. It could be allowing other materials. It's possible that also can contribute to secondary hypertension. This is just a statistical analysis of this one. As you see here, there is a really significantly higher amount of bacterial DNA in the tissue, kidney tissue, whereas in mutant, even with hypertension, relatively there is a less. It is more than the normal, but it is relatively less compared to the normal hypertensive animal. So now I will take you to different aspect. So if the bacterial components are in the organ, what are they doing? Definitely they are doing something bad to the tissue. So what are those bad things? What we think that probably they are inducing or activating the TLR4. If you remember that I told TLR4 is everywhere, and mostly they express very dominantly in the army system, meaning in the white blood cells, macrophages, and monocytes. So what we did over here, we took out the resident monocytes from the kidney. Resident monocytes means the monocytes are sitting here, like they are reserved army in the kidney. When there is a threat to the kidney, they're going to attack it. So those reserved monocytes we grind the tissue, we collected using the specific method. And what we did, we stain them with two specific markers. We have to check it whether there is a TLR4 and TLR4 activation or not, and then two, whether they are proper monocytes or not. Because if they are monocytes and if they represent or their TLR4 is activated, that means these are definitely causing some issues about there. So what we did here, this is a simple basic principle of flow cytometry I'm going to show. Probably many of you don't need to know this one, you already know, but for those who doesn't know how it works, I'm showing the basic principle, particularly for the students to understand how these techniques work. This is a sample. This is called flow. Cytoflow means you are flowing the cells through a channel, and then cyto means cells. Your cells you are pushing through a channel, and ultimately you are measuring. What it happens, we stain the cells. We collect the cells from the kidney, we stain them with our marker, expected the target marker, and then we flow through the channel. We shed light on that, and then light diffract depending on which direction it what molecules or what stains they have. It could go the straight, or it could diffract to different regions. So and then you get a pattern over here. So this is a basic principle of the flow cytometry. Using this technology, we have did flow cytometry. So what we did over here, we stained the cells with two different markers. One is CD11B+, which is monocyte mark. We have to make sure that the cells we are collecting from the kidney, these are monocytes, and then we stain them with TLR4, which is expressing whether they're expressing TLR4 or not. So we ran the cells. As you remember that they give a pattern. It's that the number here is not an issue. The number of cells you can use 10,000, 20,000, or whatever, but the pattern of cells is important. Which direction it is going. So as you see over here in normal, that's what we expected. And then we made it 4 quadrant, and you see that in the second quadrant most of the cells, these are normal. So these are definitely monocyte cells, but they are not expressing something else. But look over here in angiotensin or hypertensive normal animal, you see there are overlapping, like they are somewhere in the center and expressing in 2 quadrants over here, but we don't see in a mutant mice. So that tells us these animals not only having the monocytes, they are having monocytes, but they are also at the same time expressing or activating the TLR4, whereas these are not. That's what we expected, that there is a mutant. These are mutant cells, so that means there will not be any TLR4 activation. So that tells us that these TLR4 is getting activated in normal hypertensive condition. As you see here, there are more cells activated, monocytes, kidney monocytes in the kidney tissue. So if there is a activation and monocytes, TLR4 activation and monocytes in the kidney, what they're doing, definitely they're doing something over here, what they're doing. Probably again, I'm going back to the apoptosis. Dr. Roy already mentioned the apoptosis, but I'm talking about if there is a lot of apoptosis, we need apoptosis to clear all these trash in our body, meaning that all these dead cells and some other materials to get rid of through the vacuoles and other way. But if there is a uncontrolled apoptosis, meaning there are a lot of apoptosis, they can cause problem. They can ultimately lead to organ damage and failure. So what we see here in normal angiotensin mice, there are a huge number of dead cells in the kidney. These are kidney tissue. The blue is basically nucleus to show that these are cells, and then green you see we did a tunnel assay, so it basically gives green signals if there is a dead cells. And in mutant mice with hypertension, we see very, very less. This is a graphical statistical analysis of this data, what I have shown you over here. In normal mice with hypertension, you see huge increase, whereas in mutant mice, they are protected. So in hypertension, you see more damage, more dead cells. So if I go back, what I have shown over here, there, so basically what happens in dysbiosis condition, you have bad bacteria, increased bad bacteria in your gut. They are damaging your epithelial cells in the gut, allowing some unwanted component, including a bacterial component, to translocate to the kidney, and those components activated monocytes and TLR4 within the monocyte, and then they are causing the problem. They're killing the cells. Monocytes is engulfing probably the good cells, and therefore we see more apoptotic cells over here. So next phase, if there are more dead cells, definitely it is harming the kidney. So what harms it can cause? So we took an approach of ultrasound. Probably you are familiar with ultrasound most of the time, like when mom is expecting baby, they go to the clinic and then they do ultrasound for babies to detect baby's health and activity, whether looking healthy or not. Sometimes, it also can detect the sex of baby, but I think in India it is prohibited, but over here it is allowed. So this is funny. So this is similar technology. What we have done over here is we did ultrasound kidney. So I will show you a small video clip. What it does basically, it gives you the blood flow, how much is the velocity. It is giving peak systolic velocity and diastolic velocity. So the difference is not matter over here. What is matter over here is how fast they are going, how fast blood is flowing to the kidney and out of the kidney. That is, and through this equation, we can calculate the resistance. If there is more resistance, meaning blood is not flowing properly, that means there is a problem in the microvessels. So what we have done over here using this ultrasound method, we detected the resistive index. These are the example of each group we have done. If I present is in a graphical way, you see here. We can expect maximum resistance to 1 because it's a proportion, right? So if there is a less resistance, that means healthy or blood is flowing properly. So you see over here in normal hypertensive conditions, normal animal hypertensive conditions, you see more resistance, meaning somehow the microvessels are blocked or they are constricted, so they are not allowing proper blood flow, causing the hypertension. Meaning, so think about in a tube, you are flowing water forcefully with pressure. If you put pressure somewhere in the tube, the pressure will increase. The resistance will increase. So exactly the similar mechanism over here in the microvessels. If there's a dead cells or somehow some other damage is happening over there or already there, it could increase the resistance. That's what exactly we see here. Whether it is mutant mice or somehow to some extent is protected. You see still higher resistance than this normal one. We'll be mostly comparing these two because this is normal and this is mutant with angiotensin. These are both angiotensin. This is normal, this is mutant, and these are all saline. So we expect similar result of this one. So now we see more resistance in these mutant mice with hypertension. So if there is a resistance, definitely something is happening, remodeling in the microvessels. We know that kidney has many microvessels, including glomerular, tubules, and other things. So what we did over here, we took approach of what we call here is X-ray. This is similar mechanism. So these are soft tissue. Remember, if your bone somehow fracture, you get X-ray because bone is hard tissue. But soft tissue, we have to have a different approach. What we did over here, we infused the kidney with barium sulfate. So barium is opaque. When you put in the X-ray machine, it gives exactly similar like your bone X-ray, and it has a particular size. The minimum is I think 10 micrometer. So anything less than or anything more than or close to 10 micrometer, the barium can penetrate. Otherwise, it cannot. So if you look over here, this is a normal mice. You see nice fenestration. This is angiotensin. You see abruptly somewhere bulging, somewhere it's missing, and mutant mice very similar to like this. Here with angiotensin mutant, we also get similar like this. But here you see less venules, that less terminal arteries, meaning that somehow these terminal arteries are blocked. Remember, I showed you there is a more resistance, meaning the vessels are probably constricted. So barium is not able to penetrate further. So if we do it a reverse imaging, meaning segmental analysis, we get exactly like this. You see here there are minute vesicles in the kidney. This is the whole kidney, and we are talking about the cortical arteries because kidney has small arteries throughout the surface. So these small arteries are penetrated by the barium sulfate. Here you see mostly the big vessels, not the terminal branches. But we see these kidneys are protected, particularly this mutant mice with hypertension is protected. And this is just a statistical analysis of this to have a quick look what is going on here. You see less arteries in these normal hypertensive, but mutant hypertensive we have similar control on it. So now we see that there is more resistance, there is a less vascularity. So that means it's affecting something else. What is it affecting? Maybe there is a blood flow because the vessels are constricted. So blood flow may be less. So what we did over here, we used blood flow imaging station. So basically this is a laser technology. So we focus the laser, and this laser can penetrate up to 1 millimeter of the tissue. So we open the kidney and then focus the laser and see the blood flow, how it goes. And the results looks like this. So you can focus your target region, cortex or kidney artery or even the other tissue. You could do also in other organ, but our focus was kidney, so we looked at it. And this is the stress you get it. So if we statistically analyze those things, we get like this. So in a normal hypertensive animal, we see there is less blood flow. Basically, this technology that detects how much RBC, because RBC has hemoglobin, so basically it detects the hemoglobin passing through the light. So if there is a constriction, there is a more resistance, less blood is flowing through the surface of the kidney. That's what we exactly see. There is a less blood flow, whereas these mice are protected. In the control mutant and normal, these are similar. So now the question is, we see the constriction. We see the apoptosis. There is a more resistance in the kidney cortical arteries, and then there is less vascularity, less blood flow. That means kidney function has to be something different. What is it? How does it going on, or how to check that? We use the gold standard method. This is basically very out of the state technology, new technology, and very reliable. What we did over here, we use 5C sinistrine. So sinistrine is not decomposable within the body, and that is tagged with 5C. It's a fluorescence tag. Excuse me. So we injected this through the venule. When you inject it, it goes through the bloodstream. Okay, and if your kidney is functioning properly, kidney eliminates this 5C sinistrine very quickly, maybe within an hour or so. But if kidney is not functioning properly, what it does, it stays in the blood for a long period of time. So for example, if somebody is going through dialysis, the kidney is not functioning. So this 5C sinistrine, if you give them, it stays for days or for weeks. So that's what. When you inject it and we put a probe, this is automatic. It automatically collects how much 5C sinistrine are there. It gets basically a signal from 5C, and the computer can remotely collect all this data. This is an example how the curve looks like. So if the kidney function is properly, basically within 30 minutes, half of the 5C sinistrine can get out from the blood through the kidney. But if the kidney is somehow not working properly or malfunction, this T-half basically how long it takes to clear half of the 5C sinistrine from the body. So if the T-half shifts towards right, meaning it is taking longer time, meaning the kidney function is less. So using this technology, we analyze the data, and as you see here, GFR means glomerular filtration rate, how fast it is filtrating. In kidney, normal animal with hypertension, you see less filtration, meaning the kidney is malfunction, kidney is dysfunctional. Whereas in normal mutant or normal strain kidney, this function is normal. In mutant mice, somehow too it is protected. So if we put all these results I have shown you together, what we have seen in there over here, that during hypertension there is a dysbiosis in the gut, meaning there is a bacterial composition differences in the gut. Some of the bad bacteria is taking over, or they are exploding, and other good bacteria is basically not proliferating that level. So that leads to dysbiosis, and when the bad bacteria is there, they are basically disrupting the mucosal barrier integrity within the gut, meaning that they are damaging epithelial cells, causing the leakage of the gut, and some of the unwanted components, including bacterial components, are translocated to the kidney. When it go to kidney, it activates TLR4 and that activates also monocytes within the kidney, resident monocytes, and then these monocytes are killing the cells, ultimately leading to microvascular resistance and less vascularity, as well as kidney dysfunction through the less glomerular filtration rate. And if you somehow can block the TLR4 or TLR4 mutant mice, in our case we used, then probably we can mitigate or improve some of these bad effect within the kidney, and kidney function is protected. So with that, I would like to contribute. I would like to acknowledge many of the contributors who really did excellent job to do this study, especially Greg. He did a lot of genomic studies. Lou ran these molecular biology studies. Ruby is right now working on these apoptosis and macrophage identification. Paul was MD. He came for a few months to do some of the studies and many other things. We have excellent collaboration. Dr. Kumar has been working with me for many years, and he has medical background as well as his PhD. So he is basically bringing his medical idea to the field, and which is basically helping the entire lab to relate to the clinical studies. And David was here also. Help us to do a lot of molecular analysis and genomic code. Shabine helped me a lot for next generation sequencing. And of course, we had funding from National Institute of Health, different institute, NHLRBI and NIDDK. And Kumar was also awarded scientist development grant from American Heart Association, and all contributed. This is one of the pictures. I work here. I was very happy that Dr. Roy, our previous speaker, visited us last year, and we had a very good time over here. I work here somewhere here in this tower, half of the floor, 8th floor. This side is my laboratory, and this is another building over here. And with that, I will stop here and would be happy to answer any question that you may have. Thank you for your patience.
Thank you, Dr. Shen. Thank you for your excellent and informative presentation. We are enlightened, enriched by the excellent original work of two eminent scientists. It is a privilege and honor for me to present them in our webinar platform. Now the session is open for the audience. Now I request the participants to ask questions, if any, to Dr. Shen. You may drop your question in the chat box or may ask directly the speaker by unmuting your phone. Mona Lisa, please check whether there is any question in the chat box.
No, ma'am, there are no questions in the chat box as far as now.
Looks like that I did very bad job.
Not at all, sir.
Yes, sir. Yes. Yes, sir. They are for the previous speaker. We have already discussed about that. Okay, sir.
Okay, if no one has a question, I would ask question myself for all the audience. So this TLR4 is, we need this TLR4 for our defense mechanism. But remember Dr. Roy also mentioned, if you block one pathway, there could be another pathway. So we used only TLR4 mutant mice. In knockout mice, it could be a different story. Or like if we use inhibitors to block some of the innate immunity, then also we could get similar result. But also remember that many if you compromise your immune systems, like for cancerous patients, if you they are going under drug or cancer suppression, they are also immunocompromised, meaning that they get very quickly infected than the normal person because their immunity is suppressed. So we basically we have to balance somewhere. Some of the TLR4 is getting hyperactivated, but if you can block it or can minimize it, probably you still have a healthy life and it can have a better improved organ function, including kidney, maybe also heart or other organs.
Sir, we have one question. We just received it
Good.
just now.
At least this
At
my talk
least, sir.
gave some room to ask questions. Okay, go ahead.
Yes, sir. Okay. Sir, Priyanka asked that if increase of glucose level in blood, uh, can it damage kidney anyway?
Oh yeah, I'm glad that she asked these questions. So these two diseases, like Dr. Roy told her about the diabetes and these hypertension. These are two chronic diseases, meaning people will not die immediately, but over time, if it is uncontrolled. So it is good to have this control of this over these diseases. If it is uncontrolled over the time, what it does, it basically slowly kills the tissues or damages the tissue, ultimately leads to failure. Of course, the question she asked about the diabetes, yes, it does. Diabetic nephropathy, it is basically diabetic nephropathy or diabetic cardiomyopathy. So all these are the disease. So if it is uncontrolled, diabetes, meaning somebody is having high blood sugar, over the time, it can damages the tissue, including the whole organ. So diabetes definitely has the impact in the kidney, including other organs. And probably some of the mechanism going through this immunity, because it's when there is a excessive sugar, it is inducing inflammation. Inflammation when you get injured, so the tissue get inflamed. So inflamed tissue, if it is uncontrolled inflamed, also can kill their own body cells. So the macrophages cannot differentiate between the foreign cells and the resident cells. So it can kill all the cells. Ultimately, it can damage and kill or the dysfunction the organ. So definitely, yeah.
Thank you, sir.
Mm-hmm. Any other question?
Participants, you can ask direct question to sir if you have any.
So, can I ask a question?
Oh yeah, you can ask a lot of questions. If the organizing committee allow me to talk, I can go talk on whole day.
Yeah, I know. Very, Dr. Utpal is a very excellent work, and excellent is really amazing to see. So now I just wanted to see that when there is a leakage happens, so is it the destruction caused by these tight junctions are broken down? So is it the microbial gut microbiota which breaks it down, or what is exactly happening there?
Yeah. I'm glad that you asked. So this during dysbiosis conditions, the gut has good and bad bacteria. Both. We need good bacteria more than bad bacteria. So during disease conditions, including diabetes, hypertension, and inflammatory bowel syndrome like IBD, so there is a dysbiosis, meaning the bad bacteria is causing. So when they are basically chopping the epithelial cells, or they are ruining. Within the epithelial, there is also mucous layer. So probably so if it is uncontrolled, meaning like if you cannot control your dysbiosis, they basically slowly remove the mucosal layer of the epithelium, and then ultimately start killing or damaging the epithelial cells, including the tight junction protein. So there are different type of tight junction protein, as you know.
Right.
There are paracellular, and pericellular. So this is the occludins I talked about. Those are paracellular. So that could the other pericellular also possibly might be damaged, and if it is damaged, it is allowing all these unwanted materials from the gut to enter to the bloodstream.
Okay. So is it it is a reversible process or irreversible process? Completely irreversible.
It is a reversible possible. So if it is early on, it's early phase, then you could probably reverse. If you control in this particular case, in the bacterial composition, you could, because some of the cells will get repaired, some of the tight junction will be. It's a dynamic process. When it is healthy cells, it's a dynamic process. But if it is uncontrolled beyond the repairing phase, everything has a life. If there is a beyond the repairing phase, it cannot do anything. Then ultimately it will have a severe impact. Probably it's not curable, and it will lead to whole organ damage.
Okay, perfect. Okay, thank you. Very
No
interesting.
problem.
Very interesting.
So is there any other questions, or I can get out from here and re-enter to show my face? How does I'm looking?
I think there is no more question in the chat box.
Okay.
You can.
Okay. If there is a question, I'll come back and I will try to answer it. Okay.
So now I request Dr. Shampa Patro Madam, Assistant Professor of Zoology, Zoology Department, and Joint Organizing Secretary of this webinar, to deliver vote of thanks. Madam.
Good evening, respected Chairman, Honorable Resource Person, and my dear participants. At the outset, I must thank our guiding and inspirational force work of Dr. Manoj Ranjan Bhuiya, President of GB, and Honorable MP Rajya Sabha, along with ever so energetic and inspiring Dr. Tapon Kumar Datto, Principal Shabang Chajanikanto Mohavidyalaya, for their fatherly presence in any academic occasions of our college. It's my huge proud moment to thank Dr. Partho Roy, Professor of Biotechnology, Indian Institute of Technology, Roorkee, for his insightful views on camphorol, a flavonoid that plays critical role in protecting B cells by autophagy in a lipotoxic environment. Words are inadequate to express my gratitude to Dr. Utpal Sen, Associate Professor of Physiology, University of Louisville School of Medicine, for his lovely presentation on innate immunity, a crossroad to hypertensive nephropathy. I would also like to thank the advisory committee, coordinator of IQAC, along with the technical committee, for their wholehearted support to make this webinar happen in these toughest of times. I wish to thank all participants of Google Meet and YouTube, all faculty members and non-teaching members of our college needs a mention for their silent cooperation and support to our academic interest. This one-day international webinar on pharmacotherapeutics of diabetics and epigenetics of hypertensive nephropathy has been an intriguing session for all of us. As we are striving with COVID-19 and epidemic of diabetes, we should understand the basics of diabetes and its pathophysiology to tackle this further. Talks from these distinguished guests have added a huge amount of interest and understanding between us about the diabetes. To end with, we shall be pledging ourselves to dedicate more in the academic interest and discuss such interesting yet so necessary topics in future webinars. In these tough times, and knowledge must be infused continuously to enlighten ourselves. Thank you.
Thank you, madam, and thank all the participants again. I have some important announcement to make before the end of the session regarding e-certificate. You can download your e-certificate after 30 to 40 minutes in the same process as you join the webinar. First, you have to go to our college website, shoboncollege.ac.in webinar page. Then go to serial number 13 and tap the download e-certificate button at the top right corner, which will be active only after 30 to 40 minutes after the webinar ends. Then you fill up the feedback form. After submitting the feedback form, your certificate will be downloaded either in laptop or desktop. Thank you again. Thank you. Yes, sir. Okay, it will remain active forever. Okay, sir. Okay. Hmm. Okay.
Okay, thank you so much. Thank you for your
Thank you.
invitation. Take care and good luck to
Thank
you all.
you.
Nice
Okay,
pleasure.
thank you.
Okay, thank you. Thank you.
Sir, after
Yeah, joint organizing secretary, sir. Organizing secretary. Yes, sir. Joint organizing secretary. Yes. Okay, sir. Okay, sir. Madam, okay, sir.
Sir,
Yes, all of you. Okay.
sir.
Follow. Okay.